All right, class, let's go ahead and wrap up chapter 2 for chemistry. So now we're going to start moving on into sort of the biochemistry aspect. And the first thing that we have to discuss is the idea of organic versus inorganic molecules. Organic means that it contains carbon and hydrogen. So for instance, like a glucose molecule is C6H12O6, meaning that it contains carbon and hydrogen. And depending on the type, they will dissolve in either water or some sort of organic liquid. Now, with this being said, the water soluble organic compounds don't release ions. So, again, they're not going to ionize like what you would see with like sodium chloride. And therefore, they're considered to be non electrolytes. So, they don't release ions. And therefore, they don't also have a current flowing through them. They don't generate any sort of energy. But there are some really great examples of organic molecules. So, for instance, your carbohydrates are your sugars. So, again, when we think of like glucose, lactose, fructose, those are carbohydrates. Then we have proteins, which are going to be made up from amino acids. We have lipids, which could be like our fats or steroids. And then we have nucleic acids, such as DNA and RNA. We'll get into more detail about those later on in this chapter. Now, inorganic molecules do not contain carbon and hydrogen. Let me be careful with this though. They can contain carbon and they can contain hydrogen, but they don't contain both. So, for instance, a water molecule, H2O, is inorganic because it only contains hydrogen. It doesn't contain carbon and hydrogen. Oxygen, O2, would be inorganic because it doesn't contain either carbon and hydrogen. Carbon dioxide, CO2, is inorganic because it only contains carbon, it doesn't contain hydrogen. So they can contain carbon and they can contain hydrogen, but they have to contain both in order to be considered organic. Otherwise, they are considered to be inorganic. Inorganic molecules usually dissolve in water, and usually they can form ions. So again, when we think about ions, again, we think of like sodium and chloride. They are going to have ions associated with them. The cation would be a positive, the anion would be a negative due to those gaining and losing of electrons. And so they typically are considered electrolytes. All right, now talking about some inorganic substances really quickly. Water is the most abundant compound in living material. It's actually made up of our body weight, 2/3, and it's a major component for all of our fluids, our mediums, for our reactions within our cells, and it's very important for transporting chemicals and substances in and out of the body. You have to have water. It not only does it help with water balance, it also is going to be a solvent in which we're going to have different types of solutes dissolve in. Now, a solvent usually is and from a chemical aspect, it's the component that we see in larger quantities, and the solute is in smaller quantities. Sometimes we also think about it as what's doing the dissolving, and usually water is going to be that solvent doing the dissolving. Oxygen is also extremely important. This is used by our organelles within our cells to help with metabolic activities. And you must have oxygen for survival if you are considered to be aerobic. We are aerobic animals or organisms. Therefore, it is a necessity to have oxygen in order to release energy and to be able to carry out our metabolic activity or our day to day activity. Carbon dioxide is actually our byproduct from different catabolic or metabolic reactions. So we give off CO2, we take in oxygen, and we blow off CO2. And we have to remove excess amounts of carbon dioxide, and we do that through respiration. So again, you take a deep breath in, you're taking in oxygen, and we're going to blow out CO2. Now, we also have some very important inorganic salts. So, for instance, we do need a lot of these ions, such as sodium, chloride, potassium, and calcium. As I made mention before, if you know these different types of ions and why they're so important to our different systems, not only will you get extra credit if you send me an email, but they'll also be very important because I may have a test question that asks you about these certain guys. Sodium, for instance, is very important for our nervous system function, as well as potassium. Calcium is going to be very helpful for muscle regulation. Of course, my phone goes off right as I'm recording. Sorry, guys. But calcium is very important when it comes to muscular function and when we talk about the muscular system and helping with contraction. When we discuss other types of necessary ions like magnesium, when we discuss phosphate, they will be helpful within our bone density. And again, a lot of them also will aid in nervous system function. So if you can find out some of these important pieces of these necessary ions, again, it will be greatly beneficial for you later on. But they also play a huge role in metabolism. Again, they help with blood clotting, nervous system function, muscular processes. And again, we really have to be careful to maintain our electrolyte balance. It's all part of that homeostatic balance. And remember to revisit if you're not comfortable with the difference between positive and negative feedback. Negative feedback, again, is shutting off that initial stimulus, whether it be too high or too low. And so most of our feedback loops are going to be negative feedback. So I just wanted to kind of reiterate that and make mention of it. Now let's go into the organic substances. So we're going to start talking about macromolecules. These are going to be molecules that now are building up into something more complex. And there are 4 major macromolecules we're going to discuss. We'll talk about carbohydrates, lipids, proteins, and nucleic acids. So first, let's start with carbohydrates. Carbohydrates are what we think of when we think of our sugars. So these guys are very important when it comes to energy. They're our main source of cellular energy, but they also are going to help to build up cellular structures as well. They can be water soluble depending on their ratio of oxygen. And so again, in order to be considered organic, they have to contain carbon and hydrogen, but they're also going to contain oxygen as well. And what we see is we have a ratio of like 2 to 1. So you'll see that you have like C6H12O6, which is a glucose molecule. So you can see the ratio of hydrogen to oxygen is usually 2 to 1. I'm not going to worry about you knowing that. But I do want you to understand the different classifications of carbohydrates. So for instance, if you hear of a single sugar, a monosaccharide, mono meaning one, and that sac, thinking of sugar, these are single simple sugars, such as glucose and fructose. So that is the building block, if you will, for sugars. And we can combine 2 monosaccharides together and create a disaccharide, 2 sugars that are placed together, such as sucrose and lactose. And if you have several of these monosaccharides that are strung together, they're going to be considered a complex carbohydrate, and they make what is known as a polysaccharide. Poly meaning many, and again, that saccharide, thinking of sugar. So this is our starch, like when we think about eating breads or potatoes. We think of glycogen, which is actually our storage for glucose within our cells, and cellulose, which actually helps to make out plant cell walls. So here are just some pictures to see when you look into certain chemistry books and even biology books, you're going to see these projections for carbohydrates. Sometimes they'll show them as these straight chains like you see in figure A, and they're showing you how those covalent bonds are coming together. And just as a review, remember what a covalent bond is. It's the sharing of those electrons. And we can see that happening here. Sometimes you'll see them in a ring format. And then sometimes you'll see that there's that ring shape and they just show you a very straight structure picture of a glucose molecule. But they're all actually depicting the same thing. They're showing you that C6H12O6. Now, here what we can see is our simple sugar, that monosaccharide. So for instance, like glucose. And you can put two glucose together and form a disaccharide. And then if you put many of them together, you'll end up forming this polysaccharide. I would like you to know what they look like. So that way, again, you can identify a carbohydrate from a protein or from a DNA molecule. You won't be asked to draw it, but I would like you to know what they look like. All right, next we have lipids. So lipids, we tend to think of our fats. They don't tend to be soluble in water, so we say that they are insoluble, but they tend to be soluble in organic solvents. So again, things that contain carbon and hydrogen. And so they include our fats, which are known as triglycerides, our phospholipids, and our steroids. And they are a major component of our cell membrane, as well as they have several other functions within the cell. Now, when we think about our cell membrane, they're made up of what's called a phospholipid. And we'll discuss that a little bit more when we get to the next chapter and start talking about the cells. But they are very abundant, especially when we think of these triglycerides, which are our fats. They are also used for cellular energy. While carbohydrates are the main source, we also do have to utilize fats as well. And they actually contain more energy than carbohydrates. But what will happen is we break down sugars faster. So lipids are really more useful, especially if you need a long-term energy reserve. And like I said, what you'll end up seeing for triglycerides and how they get their name is that they have one molecule of glycerol and then they have 3 fatty acid tails. Hence why we say tri for 3, and then that glyceride is for that glycerol molecule. So that's what we tend to think about in terms of our fats. Phospholipids are similar to triglycerides, but they only have 2 fatty acid chains, and then they have 1 glycerol head, as well as they have a phosphate group, hence why they get the name phospholipids. Now, when we discuss the idea of our fatty acids, these are the fatty acid tails. These are the parts that actually make them insoluble in water. And so if they're considered to be a saturated fatty acid, that means that they only have carbon to carbon single bonds. So when we go to look at these guys, they are saturated with hydrogen atoms. And mostly these are going to have an animal fat origin associated with them, versus unsaturated fats have a double bond somewhere. And because of the fact that they have a double bond, it actually makes them liquid at room temperature, versus saturated fats tend to be solid at room temperature. And most unsaturated fatty acids are originating from some sort of plant-based diet. So when we start to look at these fatty acids for these lipids, butter would be an example of that saturated fatty acid. As you can tell from this tail, it's all carbon and hydrogen single bonds. So they're saturated with hydrogens. They're all single bonds and they're the most saturated they can be. Versus if there's a double bond in there, it can create these kinks. And those kinks allow them to be liquid at room temperature, which is why we tend to see them more in a plant derivative base. So here's a nice little picture to see of the idea of the triglyceride. So again, triglycerides are our fats. We have a glycerol portion, and then we have 3 fatty acid tails. And as you notice, the tails don't have to be the exact same type, but this is your example of a triglyceride. And like I said before, we have phospholipids. Phospholipids are very similar to triglycerides, but they have a glycerol head, 2 fatty acid tails, and then they have a phosphate group. This is a huge component, and it's the main structural component of our cell membrane. So we have these phospholipids that create a barrier between our outside and inside world. They have these, what we call polar heads, and then they have these nonpolar or hydrophobic fatty acid tails. We'll talk way more about phospholipids coming up in the next chapter when we start talking about cells. So be prepared. Phospholipids will come back for sure. Now, another example of a lipid is actually a steroid. A steroid kind of goes into its own group. And what we end up seeing is that there are 4 connected rings that are made up of carbon. And they have various functions, but they're usually used as a main component in the body in terms of cholesterol. Sometimes it can actually be used in the cell membrane to help with rigidity, to make sure that there's a little bit more structure and stability within the cell membrane. But they can also be used hormonally as well. So they're used in our adrenal glands, which are the glands right on top of the kidneys, as well as part of some of our sex hormones like testosterone and estrogen. And so here are those examples, like I said before. Here we can see the triglyceride. We have the glycerol head and the 3 fatty acid tails. Here we're going to see our phospholipid. So we have a glycerol component. We have our phosphate group, and we only have 2 fatty acid tails. And this is sort of what it ends up looking like. You have this glycerol head, this water-soluble head that likes to interact with water, and then you end up with these tails that are afraid of water. So we say they're water insoluble, and sometimes we'll say that they're hydrophobic. They have a hydrophilic head that loves to be connected to water, and then we have these hydrophobic tails. And then here's a great picture of a steroid. So again, the steroid is 4 rings that are connected with carbon. And this picture, again, I won't make you memorize necessarily what it looks like to draw it out, but I kind of just want to show you in case you take other classes. Anytime you see these points, this is indicating that a carbon is found here. So carbon is here, carbon's here, carbons here, and so on and so forth. So this is just the general structure of a steroid versus like the most abundant steroid that we see in the body is cholesterol. So we have those 4 connected rings, and then it has this side chain that actually makes it cholesterol. But we have different types as well. Again, we have testosterone, estrogen, progesterone, just to name a few. Now, the very next macromolecule, now that we've talked about carbohydrates and lipids, are proteins. Proteins are the workhorse of the cell. They are used as structural materials. They are used as transporters. They're used for energy. They're used for hormones, antibodies, and especially enzymes. And they are made up of what are known as amino acids. So I do want you to know that this is the building block for proteins. They're amino acids. And when we look at the amino acids, they have these certain groups associated with them. They have what's called an amino group, a carboxyl group, and then they have this very unique R group. I'm not going to test you on that for this time around, but an amino acid has a very specific look to it. This will come back up later on, but you don't have to know it for this upcoming test. But amino acids are going to bind together, just as we've seen before how we had the monosaccharide and they combine to create a disaccharide. We have amino acids that are going to come together and they're going to be bound by what are called peptide bonds. And they're going to make these long chains that'll eventually sort of be able to move and manipulate itself into making a protein. So this is what a typical amino acid looks like. Again, it has a general structure that it has this amine group or amino group. It has a carboxyl group. You'll see that it has a hydrogen associated with it. And then there's this funny R group assisted with it. Now, all amino acids have this component, but the R group is what makes them so special and what makes them all a little bit different. Like I said, for this test, you do not have to know that. You just have to know that the building blocks of proteins are amino acids. And like I said, they're going to come together. So this is a particular amino acid, and this is a particular amino acid, and they come together and bind via this peptide bond. This peptide bond is going to link them together, and that's what allows us to make this long chain. We call it a polypeptide chain, and it will fold its way into a specific protein. Now, when we discuss proteins, there are different levels of protein structure. We will talk about it here and again in a couple, actually next week, we'll talk about it in a little bit more detail coming up. So there's 4 levels to this protein structure. The primary level is the amino acid sequence, so how we're putting our amino acids in order. Then what's going to happen is we end up with a secondary sequence, which is going to show how they're pleated and twisted due to hydrogen bonding. If you guys remember, you have to know what hydrogen bonding is. It's due to those polar covalent bonds that it allows for this additional weak force to take place. And that's what happens in this protein structure. And then finally, we hit the tertiary, and this is where it has a unique 3D shape to it. And this really determines the function of a protein. Structure and function really relate when it comes to proteins. And then finally, we'll see a quaternary structure, which is going to be formed by some proteins, and you'll have multiple polypeptide chains connect to make just one protein. So it's very important to have these sort of levels, if you will, to actually build up our protein. And like I said, proteins can help to make hormones, they can help to make enzymes, they can help to make channels, antibodies, and all of that has to do with their shape, they're determining their function. But we can actually destroy proteins. And this is the term denaturation that you're going to hear. Denaturation is a change to either the secondary or tertiary structure of a protein. And usually it's caused by heat or a change in pH, whether it be really acidic or really alkaline. And in most cases, when denaturation takes place, it's an irreversible reaction. And because now that you've changed the shape, you've now also destroyed the function aspect to it. So denaturation can be reversible, but most times it's going to be irreversible, and it actually stops the protein from functioning correctly. So here are those 4 levels that we can see for the proteins. Again, the primary structure is just stringing together those amino acids. Those are the building blocks of the protein. And again, they're going to be strung together by those peptide bonds. In the secondary structure, we're going to see that, again, because of hydrogen bonding, they make these pleated sheets and they make these folds because of those new additional weak bonds. The tertiary structure, again, is actually going to create the polypeptide chain, and it's going to make a very unique 3D image. And again, that 3D image, that structure, is really going to dictate its function. And then sometimes, again, you'll see this quaternary structure where you have more than one protein molecule coming together to make one functional unit. All right, and then last but not least, we have nucleic acids. Nucleic acids are going to be made up of what are called nucleotides. Those are nucleic acids building blocks. And yes, I do want you to know the building blocks of these macromolecules. But the nucleic acids either contain the genetic code, like our DNA, or they can help in actually making proteins, such as what RNA does. And nucleic acids usually encode amino acid sequences. So nucleic acids and proteins kind of work together, if you will. But like I said, the nucleic acids are going to be made up of nucleotides. And the nucleotides consist of a sugar, a phosphate group, and some organic base. As I said before, there are 2 different types of nucleic acids that we'll talk about in detail. There's going to be DNA and RNA. DNA is deoxyribonucleic acid, and it's a double chain of nucleotides. And then we also have RNA, ribonucleic acid, which is going to be a single chain. DNA is going to help to carry the genetic code. RNA is going to aid in protein synthesis. And so this is what it ends up looking like when we see it. There's a phosphate group, some sort of sugar molecule, and then we'll see the base added to it as well. We're going to go into more detail about DNA and RNA coming up, as well as we'll be in much greater detail about protein synthesis. All right, so this just shows you kind of those differences. So again, DNA and RNA are made up of nucleotides. So if you string these nucleotides together, if it's double-stranded, you can form DNA. And again, we see that phosphate, the sugar, and the base. Phosphate, sugar, and the base. And these bases are particular. We're going to talk more about those bases coming up. I'm not going to spend the time here, but in next week or the following week, we'll spend a little bit more time talking about these different types of bases. But as you can see, they're actually going to be strung together and make basically a double-stranded DNA molecule. And this little piece that you're seeing here, these dots, are hydrogen bonds that are allowing for this double strand to take place. So this would be our DNA. Versus RNA is just going to be single-stranded. But again, you're going to link these nucleotides together and they can form RNA. And then again, if you link nucleotides together and then you have this double-stranded molecule, you end up with DNA. All right, so like I said, when we think about DNA, it's going to store our genetic code. So basically, it's the blueprint for everything our cells do. It's sugar in particular, because remember, it's made up of nucleotides. So it's got a sugar, a phosphate, and a base. The sugar is deoxyribose, which is why it's called deoxyribonucleic acid. And because of the fact that it is double-stranded and it has those hydrogen bonds, it ends up forming what we call a double helix structure. So again, you'll see that it's this double-stranded component. And it makes this wound up shape because of that hydrogen bonding. Versus RNA is just going to be single-stranded. It's composed of nucleotides, and its sugar is ribose, hence why it's called ribonucleic acid. And this actually interacts with the DNA to help conduct protein synthesis. And it really helps to make amino acids. So it's going to string amino acids in a particular order to help to make proteins. So as I said before, our macromolecules, proteins, and nucleic acids will really function together. And so we'll talk more detail about them coming up in a few weeks. All right, I hope you enjoyed this video. If you have any questions or concerns, feel free to reach out and ask me. Otherwise, I hope you have a wonderful day.